Okay, good. So I'm very, very happy to start this session, and I will talk about in vivo adenine base editing of PCSK9 and macaques, which reduces LDL cholesterol levels. And so I actually would like to split this whole topic, this whole project into three questions. One question is, can we permanently cure monogenic diseases? And so starting with this, I would like to quickly tell you what I understand from monogenic diseases. So they're caused by just one gene. And in our project, we actually are interested in the diseases that are just caused by a single base. And what happens is when you have in the whole DNA, like one wrong base in the whole genetic code, what can happen is then you have a dysfunctional protein. So you have the wrongly folded protein or different levels of the protein. And the solution to the whole problem would just correct the code, right? So you would just correct the base. And in our case, the problem would be familial hypercholesteremia, which leads to elevated low-density lipoprotein levels. And the solution would be adenine base editing. Just as a quick introduction, I think here in the audience, I do not have to introduce you to Cas9 nuclease, but as you all know, there's two ways of the repair of the double-strand break of the DNA, so non-homologous end joining, or also homology-directed repair. And with homology-directed repair, you would be able to say, yes, you can cure such a single base pair change. But unfortunately, this really requires dividing cells. So there was from the Leo lab, there was now really a great invention, base editors. And what they did is they used the Cas9 ligase and fused the deaminase to it. And then what happens, what you can do is, so this deaminase that deaminates the adenine to inosine. And then the inosine is red. So at the opposite strand, there's a nick. And the nick gets resected. And then this nick or the DNA is repaired using the inosine as a template. And this is read as a guanine. So you end up with a GC base pair when you started with an AG base pair. And now this big advantage here is that it works in non-dividing cells. So using this tool, our first challenge was, can we bring this thing in vivo and does it work in vivo? And we tested two different delivery methods, adeno-associated virus and lipid nanoparticles, LNPs. And for starting with adeno-associated virus, we actually tested different adenine base editors. And what we saw is that we can get very efficient editing in the liver. And actually with AB max, we get very high editing rates, 60% in the whole liver, which equals like above 80% in the primary hepatocytes. But the indel rates were lower in comparison to, for example, the ABEAD. And this actually then also results in a significant reduction of the PCSK9 levels, which also result in the reduction of the LDL levels. And then looking at the LNPs, we're actually able to really like scale this up in a dose-dependent manner. So the more you inject, the more editing you get. And also the PCSK9 levels and the LDL levels can be reduced using LNPs. What's also important here is that we can sustain this editing effect with both delivery methods for several weeks. So, okay, so it seems like, yeah, it's possible, right? You can most likely permanently cure monogenic diseases, but how safe is this approach? So when you compare AAVs with LNPs, with the AAVs, we saw that we actually have the expression. So here we co-express RFP with the base editor, and we have the expression over several weeks. We basically don't lose the base editor. The base editor keeps being expressed over several weeks. When you look at the LNPs, the story is very different. You basically have just a short period of time where the base editor is expressed, and then after 72 hours, we cannot detect it anymore. However, the editing is there. So you just need a very short period of time for the edit to occur, and then the base editor is gone again. And additionally, what we saw is when you look at the adaptive immune response, we actually saw antibodies to the base editor when we used AAVs, but we did not see any antibody response when you used LNPs in mice. Then we further looked into safety. So there was this concern that base editors might induce RNA off targets. So they would randomly deaminate adenines on RNAs. And we actually saw that in vitro as well. So if you look at the first two bars or areas here, you see the in vitro analysis, and we saw that in vitro we actually see a lot of unspecific adenine edits that we were not intended. But when you look in vivo, we actually see like far less, and only also with LNP we just see it for a short period of time, and it's gone after a week for sure. And we explain this because the dose is far lower. So in vitro we have like almost like 80% of the transcripts are actually our base editor. So obviously you would expect off targets if you highly overexpress such a base editor. But if you look in vivo, our expression levels are far lower, and this probably also explains why we see less off targets. But nevertheless, you might say, okay, RNA-seq is very noisy. There's a lot of transcripts. We might not see everything. So the more important question is actually what happens on the DNA level. So what we did here is we took the primary hepatocytes from the mice, which were edited, and then we clonally expanded a single hepatocytes. We did this because if you think about bulk sequencing and there would be an off-target that is very rare, you would not find it in the noise of all the other DNA from other cells. And if you would do single-cell sequencing, you also have a high error rate by the method itself, so you would also not find it. So we thought we just exploit nature itself, and we take the cell, let it expand, make accurate copies of its DNA, and then do whole genome sequencing. And what you then get is a so-called 96 nucleotide graph from each individual clone, where you see the different conversion rates within the whole genome and the DNA that we find. And if you look at the fifth column here, you see the T to C conversions, which is also including the A to G conversions, which are the conversions that should be done or should be found when you use adenine base editor. And when we now compare this to control clones, AAV clones, and LNP clones, we actually did not see any increase of these edits. So we could not find any edits on a DNA level. And then to even further prove the safety, we came up with a long-time study in a sensitized mouse model. So we had mice that had a knockout of P53, so the major tumor suppressor gene, in the hepatocytes, and we confirmed this, that the P53 is really gone. We injected them with the base editor, so we made sure that it also worked, the base editor was in there by checking the PCSK9 levels. And we also checked the editing in the end. And they really had, for over a year, they had base editor in. And then we looked at the livers, and we even gave the samples to a pathologist, and we really did not find any signs of any cancer formation. So this long-term study even further seemed to show that there cannot be a lot of off-targets happening that would be very detrimental. Okay, so we think the approach might be pretty safe. So now the next question was, can we translate our findings? So would it be feasible for clinics? And for this, we actually were able, so I have to thank here already Acuitas Therapeutics, because without them, it would not have been possible to do this big non-human primate study. And here we tested with the LNPs whether or not we can have efficient editing in a large animal model. And what we saw is that we actually achieve it. So using 1.5 mg per kg, we actually have editing rates of around 30%. So these editing rates are high enough to significantly lower PCSK9 and also LDL levels. And then we further looked into the innate immune response and also liver toxicity. And what we saw is like right after injection, so if you look at the gray and the dark yellow bar, these animals got a second dose. And every time after injection, you got like a very quick increase, but it goes back to baseline very quickly. So this again speaks for the LNP approach, because it is so transient that you just need it for a short period of time, but then after editing it happened, you don't see any side effects anymore. We also here looked at adaptive immune response, and interestingly, actually here with the LNP approach in comparison to the mouse approach, we saw adaptive immune response to the base editor. So this underlines furthermore that a large animal experiment is important, because the immune response in larger animals is probably more sensitive in comparison to rodents. So yeah, so altogether, I think I could show you that it probably is possible to permanently cure monogenic diseases. I would think it's a pretty safe approach in comparison to other approaches out there yet. And I think it's also possible to scale it up, and theoretically it would be feasible for the clinics. And with this, I would like to already acknowledge the people that were helping. So I would like to thank Melissa, Paulo, Ying, and Sean for the help there from Macaques Therapeutics and made this big study possible. And also would like to thank the people from Syntago that helped with the production and also the large and also the small scale experiments with providing the guide RNAs and different modifications. I would like to thank the people that did the bioinformatic analysis and also the people from my lab. And first and foremost, don't want to forget to thank Gerard. He is my PhD supervisor, and with him, obviously, without him, it would not have been possible. And yes, I would also like to thank the funding and you for listening, and I would be happy to answer any questions.
Well, you received a whole pile of questions. I know you're probably too many to scroll through in the short amount of time. One of the questions was on utility for primary cells and such. I think you've got some believers that hope to jump on this.
Sorry, it's in the public. Can you repeat it? I think I don't see them.
Oh, one of the. Okay, so I will look to try and transfer them over. But the question was, is it possible to use in primary cells? Was one of the early questions that came in.
Oh, yeah. Okay, yeah. So actually that's something that we had to overcome because we could not expand primary hepatocytes. We actually had to use a protocol that was published by the first author is Katsuda, and they it's a cell paper, and they show that you can convert them into liver progenitors. They call it chemical-induced liver progenitors. And you give them a cocktail, so you actually have an intermediate stage. It's not really stem cells, but it is like a progenitor cell from the hepatocytes. And in this stage, you can actually expand them. Yes.
And so the second question, which is probably all we have time for, is you show one set of data, and now they want to know what's the consistency between batches.
I don't know what we are actually talking about. I assume it's probably the whole genome sequencing and the RNA-seq data where we have different batches. So we always took different animals when we compared. So then the variations, for example, for the RNA-seq was very similar, even though it was different animals. So we found like similar amounts, even though it was different off-targets, we still saw a similar off-target amounts that we could detect.
I guess the question was in terms of the LNP and such. The question was how consistent was the final product?
Okay, I'm sorry for. Okay, thank you for this clarification. Yes, no, that actually with the LNPs, so we tested also different guide RNAs. So we always had like the same mRNA of the base editor, but then we also complexed it with different guide RNAs, and we really got it very robust. So the animals were also not injected always at the same day, and we also did the same experiment sometimes at another day, and we could observe the same result. Absolutely, it was very, very reproducible working with the LNP.
Wonderful. Thank you so much.